So I would start this short intro on liquid biopsy, where are we right now. And I think just for you, this was really the concept that was introduced by Katja and me 15 years ago for the analysis of circulating tumor cells. And now many people just use it for the circulating DNA and for nothing else. But I think we do feel that liquid biopsy is really encompassing not only CTCs and not only ctDNAs, but also other circulating biomarkers such as RMAs, extracellular vesicles, metabolites, and other things. And it may also encompass circulating host cells, which of course play an important role in the induction of tumors, but also in the progression of tumors. So maybe sometimes that's something we forgot. The field is very tumor-centric, right? We do want to analyze everything that's derived from the tumor cells, but of course the tissue microenvironment and the host environment could be also very important, maybe even important to detect cancer. So I think that all of you are convinced that the blood, of course, has some advantages. Tissue biopsies are still, of course, the key in cancer diagnosis, but for early cancer detection, there is, of course, no tissue available, so we rely on something else. Blood, of course, is super minimal invasive. Even some people faint after a blood draw, but I guess that's why it's called minimal invasive and not non-invasive. I had to learn this difference. But what is very important is that blood also, of course, is a pool of tumor cells and tumor cell products. And I think that has maybe the advantage if we get more comprehensive information. And of course, we can take blood samples over time. And even for early cancer detection, I think people like Dennis Lowe and others had shown that if you take maybe 2 blood samples, you could increase your sensitivity of detection. And so maybe even that could apply to early cancer detection. This has been actually highlighted very nicely in the 2020 Nature Medicine edition by Jana Romero. I also like to show this slide because this Nature Reviews Clinical Oncology is really a nice oncology paper, a nice review paper, especially for the young people. If you want to get some idea beyond your own PhD thesis, these review journals really give a very comprehensive, usually very well-written introduction into other fields, and I can really highly encourage you to look at these review journals. There's also Nature Reviews Cancers and others that really may give you a very quick but very, very well defined introduction into new fields. Well, of course, when we want to detect cancer and in the blood, we can have different sources. We are interested very much here actually in, can we detect the primary lesion early enough? For example, in our case, primary pancreatic cancer. But of course, the tumor can progress, and if we have lymph node metastasis, they can also contribute into the blood. And if we have already distant metastasis, this could be also a big contributor to the liquid biopsy biomarker. So depending on the stage of disease, different sources can actually contribute. And in the blood, and I have already highlighted that in the slide before, you have, of course, the cellular fraction. And here we actually work a lot on the circulating tumor cells for a long time. And Kati will focus on that more. We have single cells clusters, but we also have the circulating immune cells here, and the endothelial cells, and also the circulating fibroblasts. And they can also contribute. And in the plasma and serum fraction, we have, of course, the circulating DNA, which is a major biomarker in our consortium, but also the circulating EBs, protein, RNAs, and EB tumor-educated platelets. I just like to show this slide. It's unpublished work. We have really looked here at protein composition, or EVs, in actually lung cancer patients. And usually when we do our cancer studies, we do cancer controls, right? We say we have the controls, healthy controls, and we have the cancer patients. But who the hell is healthy, And controls, look at us here. I mean, there's nobody here who looks like the other, right? And everybody has probably some diseases that the other doesn't have and some habits. And one of the habits that affects biomarker constitution largely is really smoking. And that's shown here on this slide. We just detected here more than 4,000 proteins from CEVs of 1.5 ml of human blood plasma. And in red, these are the healthy, I don't know whether they're healthy, the control smokers. And then you have the healthy control non-smokers. And you see here in this Venn diagram beautifully, you can separate these two groups. None of these individuals had cancer, okay? And I think we found almost 300 differentially expressed proteins between these two groups. So it's also something we have to keep in mind if we are really looking for cancer control studies. I mean, we have to have a look also what kind of controls, what are we really comparing? Otherwise, we get signatures for something else than cancer. Okay, I also should say that in the circulation, as I said, we had the circulating tumor cells, but we also have the non-tumor cells. Here, of course, we detected circulating the fibroblasts. They are derived from the tumor microenvironment, but they may also be derived from other sources. So people call them cups. We do that too, but of course, in the circulation, you're not totally clear. We have found some phenotypes that were very low in healthy controls, but I guess that is what was really obvious, and it's shown here. The pointer is not really very helpful. There you have many more circulating cups than you have circulating tumor cells, so it's a biomarker with a higher dynamic range. Of course, for the circulating tumor cells, you can also get genetic information, and people would say, why do you need to do that? We can do that from the circulating DNA. But maybe one of the chances with the circulating tumor cells is that you can look at heterogeneity. You know, in medicine, we always want to say, okay, this patient has a targetable genetic mutation. For example, these are melanoma patients, and BRAF mutations are targetable with drugs. And you would say this patient is positive or is negative. But all of you probably know that positive does not really exist in that way, that all the cells must be positive. There can be tumor heterogeneity. And for all the solid tumors, heterogeneity is the hallmark, unfortunately, because that may also explain why we cannot cure these tumors so effectively. And that's shown here really. We analyzed 7 circulating tumor cells from these melanoma patients, and we could find BRAF mutations in 5 out of 7 cells. The other one were wild type. So you had actually a chimera in a certain way. Okay, well, coming now to the ctDNA, I think that all of you know that the major source are dying tumor cells, but there is also some circulating DNA encased in the HBs. And of course, we can use all kinds of sophisticated genetic analysis to prove which fraction of the circulating DNA is derived from the tumor, because we also have a sea of normal DNA. So that's why we need sensitive methods. So what are the methods in principle? I think we have heard about that already many times. We have in principle tumor-informed and tumor-non-informed assays, also sometimes called tumor-agnostic assays. For the tumor-informed, you need to get information on the individual composition of the patient from usually the primary tumors. So you have a very specific information, and you can really then design tests with a very high sensitivity. For the tumor non-informed, the advantage here is you don't need tumor tissue, which sometimes is difficult. For example, we have a lung cancer study where we have in 20 to 30% of the cases biopsies that have not enough quality for whole genome sequencing. So these real-world problems can sometimes also dictate what kind of tests will make it in the future. That's why there's also quite some interest now in tumor non-informed tests. And of course, in early cancer detection, you don't know what kind of tumor you have. So you have to go for a tumor non-informed test in that regard. So what are the challenges? I think all of you probably know that you have cancer-associated mutations in leukocytes, these SHIP mutations, which are also responsible for all kinds of predilection to diseases. They have become super interesting as subject of investigation. So please, if you do a review on SHIP mutations, have a look at it. It's very interesting. but also other normal-looking cells, such as normal-looking breast cancer cells or bladder epithelial cells, have already cancer-associated mutations, even if there's no cancer detectable by any pathologist in the world. So you may have a background. So I think, on the other hand, you may also have tumor lesions that are non-shedders, and we still don't understand enough about the biology of the shedding process. So if you have a chance in your PhD thesis to really go into that a bit deeper, I think that Yuval and I would be super happy, because we still are old-fashioned and believe that understanding the biology will lead to better methods of clinical applications. So, and of course, we all know that if we want to detect early-stage pancreatic cancer or other tumors, we need to detect very low concentrations of analytes. That's probably not only true for the DNA, but also for the others. So this just gives you an idea what really sensitivity is probably needed. We here did a study, it's also not published yet, on melanoma, and we used a method that went down to 2 parts per million, a tumor-informed method. We can detect the BRAF mutation, as you can see here, very nicely, but we can also detect all kinds of rarer mutations with such an ultra-sensitive method. And if the patients were negative doing the monitoring, they had a much better prognosis. You know these Kaplan-Meier curves, all patients alike at the beginning of the observation and during the time. Patients that were positive died, and patients that were negative survived. But the more interesting is really that almost 30% of the patients had ctDNA levels below 100 parts per million, so very low levels. Also, these were metastatic melanoma patients, so we're really dealing with very low concentrations. So, just to give you a glimpse in the clinical applications in the next 10 minutes, I think that we are here in PANCAID. We want to detect tumors and aggressive tumors, pancreatic cancer, super aggressive tumor, as early as possible. But of course, other applications include to get a better risk assessment, which patients will relapse, then even detect minimal residual disease, then monitor the therapy based on the findings, and in the advanced setting also find out what kind of resistant mechanisms are really available or have really been developed. And if we are lucky, we have drugs against these resistant mechanisms. Early detection of cancer, I think this was one of many studies that has received non-op attention, where the group used actually methylation-based analysis of ctDNA, and they developed a pan-cancer test based on that. And you see here, yes, they were able to detect a series of solid tumors, including RIS1 pancreatic cancer, stage 2, and they were also able to detect a lot of blood cancers. But I think that in less than 50%, these were early stage 1 or 2. So I think there's room for improvement. That was probably also the reason why the EU had set up this early detection call, which I think was totally overstriped. We are so lucky. Only 6 out of 87 projects were actually funded. So that was one of the lowest funding rates in EU history. I saw the statistics. Usually it's like 15 to 30%. So we can be really lucky, and it's also a lot of pressure on us to really do well. So you all know the PANCAID project. I don't have to really go deeply into it. We are now in the midterm, and I think we will hear a lot about that. What I always think is really great, we have a very good biobank that goes back many years and also includes pre-cancer lesions. We have experts from all the liquid biopsy fields, and we have with Nui and their group a very fantastic bioinformatics group. And what I think is also really great here, we have the health economics. That's quite rare in EU projects, especially projects that are led by a wonderful clinical collaborator and laboratory guy who really include also other species, so to say. But that's super important for screening projects. Ethics is also super important to understand what is really ethically welcome. And of course, we have the great dissemination management from Concentrus here. So the next application is detection of minimal residual disease. It's a bit like detection of early cancer. It's actually not so different. The advantage here, you know the enemy. Okay, you know whether somebody has a breast cancer, pancreatic cancer, or lung cancer, which makes it a bit easier. But again, we have to detect very small amounts. And this, of course, gives now the chance to have here a signal that the tumor may come back between the early detection and the relapse. And that gives us also a chance, really, and the companies that develop drugs, a chance to here design a new way of post-adjuvant therapies that may block the relapse of the cancer. And I don't want to go into many details. We have another EU project on that. It's a guide MRD, and several of you are also part of that. And here we create a reference standard and benchmark these DNA assays that are available right now commercially. We then do robust validation in colorectal, pancreatic, lung cancer, and then we want to use that to guide treatment. And in this EU project, that's also interesting, it's called an IHI initiative. You have the partnership of academic partner and companies. And it's almost a 50-50 funding. The EU gives you 50% and the companies give you 50%. If the companies give you a lot of money, the EU gives you a lot of money. Huh? So it's an interesting interaction, I think, for implementation. I won't want to get into any details here. This is just to let you know, if you go in early cancer relapse detection or MRD detection, and it might be not so different for early cancer detection, we always want to measure the tumor burden. And we are, of course, happy if we can see that the treatment did reduce the tumor burden. And then we feel that there's an exponential regrowth in some way, a quick regrowth or a very late regrowth. But I guess there's also the host control that is very important that we may overlook sometimes. It's not just that you have probably breast cancer patients that had the same tumor load, the residual tumor load, but one of them can control it and the other cannot control it so well. So you may have a different curve of relapse, depending not only on the tumor burden, but also on the host control. And I just can go over that quickly. And I think that another question really in the field is, whenever you want to treat metastatic relapse, you take the primary tumor from the pathology biobank. But in some tumors, not in pancreatic cancer maybe, but in other tumors with a longer latency period, this can be material that is 5 years old, and the patients in between had chemotherapy, immunotherapy, whatever. So there's a lot of pressure. So is it really good enough to just take the primary tumor, or has the metastatic tumor shifted into something else? And that was done here with HER2, which is a very common target. We had seen that in the case of breast cancer, when the patient become metastatic, 10% to 15% of the patients had switched from a HER2 negative to HER2 positive status. And we could analyze that by looking at the HER2 status of the circulating tumor cells. So we said, okay, let's design a trial, multi-center randomized, where we treat patients that were originally negative. They never would get an anti-HER2 therapy because the primary tumor was negative, but they were now positive in the metastatic situation. And as you can see here, the red curves, the patients that received additional HER2 therapy had a better overall survival than the patients that just got the standard therapy. And of course, there are many, many other, I think, targets, I think that are either can be detected on the genomic level or on the protein level. So that's why we feel that the ctDNA and the CTCs are really complementary. It's not really a competition. And for the, of course, gene analysis, I mean, below in lung cancer, mutations, EGFR, in breast cancer, resistance mutations in the estrogen receptor gene, and now in prostate cancer, mutations in the androgen receptor ligand binding gene, which will become important because there are drugs available that can be then applied. And I think we also did a study with such a limited set of genes, the tumor agnostic set, where we target 5 genes in melanoma patients. And that's just to show you here, this was the mutational spectrum before therapy at baseline, and that was the spectrum during the immunotherapy. And even from the last row, you may see that, yes, there are really some dynamics going on. So the spectrum of gene mutations in the ctDNA changed during therapy. And that's also the beauty of liquid biopsies, that we are able really to monitor that. Another question is, can we detect the tumor of origin? I mean, if we have a pan-cancer test, for example, it is the horror of all healthcare institutions worldwide that you say, okay, you have a tumor, I don't know where. And then you have to do, I don't know, a number of CT scans, MRT scans, and maybe you don't find it because whether you have a tumor in the brain or a tumor in the prostate, it's quite a different procedure of detection. So this would be extremely valuable for a cancer screening test to say, okay, this is really a specific type of tumor that we are looking at. And here we had a program going on with funded by the German Cancer Aid, where we look at patients of cancer of unknown primary. These are patients where you first detect a metastasis, no primary tumor can be detected. So the primary tumor has probably not grown or has been regressed, or nobody knows exactly why you detect just the metastasis first. And one of the questions is, what is the origin of this metastasis? Because the therapy of a brain tumor or prostate tumor is totally different, right? So what we had done here, we had looked at circulating tumor cells where we could see, okay, if you have those cells, the prognosis is worse than if you don't have those cells or less of those cells. But what is now interesting, we are now looking at ctDNA with the methylation profile. And we have now, I think, around 70% of the patients where we can get tumor of origin information. And that has been just presented at the ESMO as a CDAC. Clinical implementation is super important. We all know that everybody in the beginning of our careers, we believe if you publish a great paper, and we have here colleagues who have published wonderful papers in super journals, right? You feel, wow, then it should be taken up by somebody and be implemented in the clinic tomorrow, okay? If you get a bit older like me, you understand, no, it doesn't work this way, and you have 10,000 of publication in the biomarker field and not even 10 new biomarkers in that clinical implementation. So there's an implementation gap, which has become very important now in Brussels. We are now in Brussels at the EU. The EU is now understanding. So this implementation gap is a bit dangerous, because you can imagine there are people at the EU who want to give the money not for cancer research, but for other purposes. I don't have to say which purposes, right? I mean, it's so obvious. There are so many people who need money, okay? And these people now come and say, look at these researchers. They get all these millions and millions from us just to publish papers and become professors, okay? So we better take this money away from them. So this implementation gap is now really important to overcome. It's not just because we want to have it. We want to, of course, do it for the patients. I mean, it's our ethical thing, right? But even beyond this ethical thing, I think it would be really clever if we could implement things to really prove that what we are doing is also good for society and not for a bubble of academic work. So we have really, and I don't want to go into details, after a EU project established this network because we want to sustain also what's going on. After 5 years, we have to see what can we do. So we went on with it, and everybody gives a little contribution here each year. And we meet once a year, and we have many, many activities. We have working groups on the clinical questions, on dissemination and education, which is very important. And Kati is leading it together with Cole Hoffman. Clinical, of course, to say what are the clinical questions of relevance. Technology is what technology can really address these questions. Regulatory working group is about IVDR and reimbursement. Data computation, super important, of course, to bring data together. And we have a new patient advocacy group that you want to establish. And I think the wonderful colleagues here from PCE, I think I want to become very active in that, and I'm super happy to do that. And if you're interested, we are now already over 100 institutions. It's an old slide because it's really growing fastly. Just send me a mail and let me know if you want to participate. And we also have published our activities here and in this journal. It was at our last General Assembly. So if you want to have a look what's going on in the society, please go into this publication. So at the end, I think we have really, with Liquid Biopsy, the chance to go from early cancer detection, over risk assessment, MRD detection, treatment selection, response assessment, to the identification of resistant mechanisms. So it's, I think we can follow the patients through the whole journey, which I think is fantastic. And I want to thank my colleagues here in Hamburg, and also the EU contribution, which was really fantastic for our work. And these are the new two members of our team, my grandsons, Emil and Frederick, who will start to pipette soon if they don't eat the tips. So thank you.
So I'm very happy to be here today and to talk about liquid biopsy for sure. But I decided to focus more on the biology of the metastatic cascade because I feel that it's very important to understand the biology of this process to better maybe detect the different biomarkers and also to be able to target new, I would say, proteins or whatever to find new drugs. So that's why I will try to talk about that. So if you see the different steps, I would say, from the metastatic cascade, you see that you have really different key identified steps here. And of course, what we are interested more is really what is going on in the dissemination of the disease, tumor dissemination. And that's why it's really where we would like to understand better what is going on. But you see that you have all these different steps. And of course, it is really a clonal selection. That's why it's an inefficient, I would say, process, which is always said by Hans Chambers for many years, who worked in Toronto. So when we ask, is there one key step in the metastatic cascade, with my colleague, biologist of evolution in Montpellier, Frédéric Thomas, we looked with a mathematical model and we tried to see all the different steps. And when we tried to decrease all of them, the only one which was very important was the survival duration of CTCs in the bloodstream, because it's CTCs. So you see that it's really important to focus on that specific step, because when we try to affect the other steps, it was not really important for the clinical development of metastasis. That's only the CTC survival, which made sense to try to decrease it and to understand how it works. So that's why I would like now to focus on that. So it's a new review that we just published with Klaus, and that's why I would like to dissect it one by one. So first, you may know that they are not really attached to the basal membrane anymore. So it means that they need to survive this specific apoptosis, which is anoikis. So it's really important to know that it's really important for CTCs. Then, of course, you have this huge heterogeneity. You see, you have heterogeneity because you see on the left side that you have like small cells, big cells, different plasticity for the deformability as well. You can see on the right side that you have this heterogeneity with mutations at the transcriptional level, but also at the proteome level, secretome. So it's like full information that we can get from a single cell. And of course, you see that I put stainless because we know that a very small subset of these TTCs is able to self-renew and to give rise to metastases. And many people don't remember, but in 99.9% of the cases, for example, in breast, colon, and prostate cancer, the patients will die from the metastatic sites and not from the primary tumor. So that's why it's really important to try to block this tumor dissemination and to try to understand which are these stemness plus CTCs. Then you see plasticity. I will also give you examples, but plasticity is very important because we know today that you have this transition. So this transition that you can have from epithelial to mesenchymal state. because we have like a few epithelial cells, and then they can have the ability to change at the morphological level. So they are more mobile. They can change also what they express. That's why it's important to detect CTCs, but also EVs. So it's quite important to go on that topic as well. And finally, I would like to say that more you are close to the tumor, better you can detect your elements, because you have a clearance of the different circulating tumor cells and also DNA. But if you are close to the tumor, so for example, in the draining vein, you have a higher chance to detect these circulating biomarkers. So where to perform liquid biopsy? A few years ago, we tried to see in the literature, what is really important to do when you are close to the tumor, the draining veins. So it was clear in colorectal, pancreatic, liver, and lung cancer that if you are in the draining vein, you are really more CTCs, for example. So we did that, you see, a long time ago when we had a function in the mesenteric vein and in the peripheral blood, and we could show that you have a very huge gradient from close to the tumor before the liver and after the liver in the ham vein. So it's quite clear that the liver is on the way for sure, but it's also a place where the CTCs are very much attracted, and they can develop, of course, very nicely metastases there. So you see that it was already done in colorectal cancer. More recently, we did the same in pancreatic cancer, where we could show as well with our colleagues in Bordeaux that you have like localized pancreatic cancer patients. And then when we did the peripheral blood, we could not find in this specific patient 0 CTCs, but you see that in the portal vein, we could show that we have a lot of CTCs and even clusters here. And of course, without mobilizing the tumor, because if you touch the tumor, you try to do mechanically something, of course, you have a huge release during surgery of tumor cells, but it's from primary tumor cells, not CTCs. CTCs, the definition is really that intrinsically or even from the microenvironment, you have a signal to have a release of the more aggressive cells. And if you do that during surgery, of course, you have a peak of CTCs, not CTCs, tumor cells, but that's why it's very important to do these experiments while you have not really touched the tumor at all before surgery. So another way to do it is not to go, for example, close to the tumor, but close to the metastases. And it's also another work that we did with Amélie Darlix, medical oncologist in Montpellier, where we looked at the cerebrospinal fluids for breast cancer patients with leptomeningeal metastases. And you see here also that it was quite nice to detect the CTCs in the CSF, and you have also information directly of the phenotype of these CTCs. And I remember that in this specific patient, it was completely different in the blood. For example, even the phenotype was completely HER2 negative, and here we had 16% of the patients of the CTC that were HER2 amplified. So very much important that the information was coming from the metastases, and it was of course different from the blood, from maybe the primary sites. And what we published here, we compare to what we are doing in the routine clinical practice. And of course, we could get more information more quickly and on more patients. So the question is how to do next for this analysis in the future, and to get information about HER2 from the metastases and not from what was at the beginning of the disease. So it's what we have seen now, the different, I would say, CT3 hallmarks. But now I would like to go on with something that I feel very important as well, is, as you know, ctDNA and CTCs, they are in the blood for a very short time, like 15 minutes to 1-2 hours maximum. So it means that you have a very short survival, which is nice because you can say, okay, it's a problem. I feel that it's a positive point because it's not the accumulation of information of mutations, phenotypes, and so on for the last 6 months or few weeks. You have really the last emerging clones information, which is completely amazing. So compared to, for example, a tissue biopsy where it's static, you may go to a certain place, but you miss some minor clones. So here it's really what is released from the primary site, but also distant sites during a very short time. So you know exactly what is going on real time. So that's important. But now the question is, what are these survivors from not really dying in a short time? And it's what I said, a very rare subset of CTCs able to give rise to distant metastases. And that's why we are very much interested in that specific subclone, because you can imagine that if we understand them better, we can kill them better, and we can for sure block maybe the next step of the metastasis development. So, of course, the involvement of immune escape, we are sure that there is a crosstalk with blood cells and immune cells, which is very important. But also we are studying these days as well the size selection. Very important to know that of course, if you have microcapillaries, you can imagine that these cells can be blocked somewhere, and you have also naturally a selection of the CTCs based on the size. You have the shear force resistance, and of course, the notion of cancer dormancy that we heard already by Klaus, but which is very important in, for sure, breast cancer, where it's very knowing. So let's start with immuno-oncology and I would say the immune escape, which I feel that is very important. And as Klaus said just before, that it was ignored the last years. In the liquid biopsy, you have access to, of course, what is derived from the tumor, but what is induced as well by the tumor is very important. As Klaus said, you can imagine that a weak immune system will let the space to the development of cancer. And if you have a strong one, of course, it will be different, so the progression. So very important to have that. But first, if you have a CTC going in the blood, you will imagine that it should be eradicated very quickly. And for some of them, it will be not the case. So that's why you have this interaction. You see these many different blood cells, even the platelets that can help the cells in the blood to roll on the endothelial cells and to extravasate at the right place, at the right organs. For example, we know in breast, it is liver, lung, brain, and bone marrow. So that's why we are now also working on this specific aspect. So for example, CTCs we know can release inhibitory cytokines, so they are not attacked by the natural killer. And if you have some interest in that, Dorian Masboudi, a PhD student in my lab, really wrote a very nice a review on that. CTCs and natural killer. We need to understand how they are cross-talking. Then, of course, you have this do not eat me signal, CD47. Then the macrophages are not killing specifically the CTCs. I just mentioned also the platelets, where you can have the CTCs completely covered by platelets. And you see that you have the secretion by the platelets of TGF-beta, which is nice because they are maintaining the CTCs in this EMT state. So that's why they are helping also to travel in a good condition. And it's really Zara Eslami that some of you know very well. She was a PhD and now she's still in my lab, and she had a very nice work on the crosstalk between CTCs and platelets. And she could really show very nicely that, of course, you have first the CTCs will cause phenotypic changes, activation, and so on in platelets. So we have tumor-educated platelets, even early enough in the megakaryocytes. But you see on the other side that you have also the increase of the invasive properties of CTCs because the platelets are helping them to surviving the bloods. So it's really nice to see how they are interacting together. And I met, of course, the last PDL1 expression on CTCs, that it's like a camouflage, and you know how the CTCs are not, like the tumor cells, destroyed anymore by the immune system. And I would like to say that we first demonstrated the expression of PD-L1 on CTCs 3 years before the Nobel Prize was given to Onjo Tatsupu and Jen Sadison in 2018. So it was really very nice to show that in metastatic breast cancer. Then, of course, we had to show that it was clinically relevant compared to tissue biopsy. I felt that it was not only important to detect them, but to show the relevance for the patients. And also, more recently, we had the same approach for non-small cell lung cancer. And just to show you here that it was in the context of chemotherapy, because at this time, immunotherapy was not given, as I just mentioned. So now we have new project on, of course, immunotherapy and to detect not only CTCs, but others. And why? Because when we have this very nice work with Jessica Gobu also working on EVs, we looked at EVs expressing PD-L1 and CTCs expressing PD-L1. And the winning combination was really the combination of CTC and EVs PD-L1 plus. So you see that it's nice to go towards a combination of circulating biomarkers. So I just mentioned PD-L1, but what we have looked as well, IXL expression, because we know that you have more and more therapies against IXL on tumor cells. And it was important to show that we have an expression of IXL on certain CTCs in some patients. But now we are, of course, following these patients to see whether there is also a clinical relevance or not. So that's the next steps, I would say. And what Dorian is doing, I feel that it's important as well, is to see what is really at the surface of the CTCs, what is really interacting with the immune cells or even the blood cells. So what we are doing is really the surfacome investigation, because we want to know how the CTCs can evade the immune system attack, but also we want to develop maybe new approaches because we will discover new proteins expressed at the surface. And the project is going very well. He's analyzing all the proteomic analyses now. And of course, the idea is to find something else than PD-L1, because you know that you have like 30% of the patients responding to this therapy. And what about the 70% left on the side? So we need to have more than PD-L1 in the future, I feel. So that's why it's really important, and not to work on just tumor cells like that, but we have the chance to have CTC lyes, which means that we are working on CTC lyes, the ones that are giving rise to distant metastases, the ones that we need to block and to eradicate. So we worked a very nice review on that, the approach and the technology. So the idea is really to do the glycoproteomics, but also maybe if we have time, the immunopeptidomics. What is really in the interaction with the immune system. So with the help, of course, of our colleagues at the platform of Cokeonics, and we had to really implement a new protocol for CTCs. And for that, I could get a huge French project on cyanids for signature in immunotherapy. So we will use liquid biopsy to try to see which patient should benefit of immunotherapy and then whether the immunotherapy is effective. And second, we have another one, Transcan, Equilunar, financed by the INCA this time. And the coordinator here is Angel Lagares in Spain. So what about tumor dormancy? Because Klaus mentioned briefly that, and I feel that it's very important as well, because you may have some cells really at the single cell level completely hidden somewhere in different organs, but maybe mainly in the bone marrow as a reservoir of these disseminating tumor cells. And of course, it's linked to what? To MRD detection, because we need to understand how these cells can be protected in some organs when they are already, again, producing some, I would say, DNA in the blood, because you have like maybe a more replication of these cells than a higher release of DNA in the blood, or even single CTCs that can also be released. And you see that it's very important to understand this process, and many groups are working on that. And that's why, again, the role of the immune system is crucial. Of course, the microenvironment, but also what is blocking and maintaining this dormancy here. And at one moment, of course, you have this awaken of these cells, and you see on the right side, you have still some questions for the biology of MRD. How does ctDNA accumulate in the body fluids of patients with cancer? Why are malignant cells actively released by the tumor, and at what point during cancer development? And also during our last meeting in Harrows with Carol Lees, we also questioned what is the shedding of CTCs and ctDNA and the clearance, because then you may have, of course, at the end, something that you don't see anymore, or maybe that is now visible. So that's why it's really important, again, to go back to the biology of what is going on for the cancer dormancy. And Klaus mentioned, of course, that he's leading this very important guide MRD consortium with Klaus Andersen. And again, when we look at that, what is Dickmeyer? We need to identify these small traces of residual cancer post-surgery, for example. And if you do that early enough, you can imagine that you have a better treatment of these patients, because you can really do the prediction, of course, but you can also personalize the treatment based on MRD. So very important. And again, I don't talk about that today, about the technology, but it is like it has been said before, you know, you need very sensitive and specific methods to be early enough and to be specific and not to see like confounding biomarkers. So we have seen everything here on the left and middle side. What I would like to say as well, that the CTCs are disseminating, but they are not going everywhere like that. They are going to specific places, which is quite interesting as well from the biology point of view, because they will be attracted to by specific organs, maybe chemoattractants, but we know also that you have this exosome dissemination very early during the tumor development, very early to the preparation of the pre-metastatic niches, which is quite amazing as well, that it's more a systemic disease than a localized disease. So you have the release of exosomes or extracellular vesicles very early to prepare the pre-metastatic niches, and it's all the work also from David Leiden, and to show that you have this exosome going to specific places. And then when a CTC is arriving, of course, we know the soil and the seed can grow. So it's quite interesting to see that. And that's why I believe for early detection that it's very important to go to that step, because it's early enough to be maybe detectable. And at the end, I would like to show also when to do a liquid biopsy, and maybe it's something that you never thought of it, when to do that, because when the COVID was there, I asked my students to say, okay, do a malignant cell skip at night. And then we worked on a review on that specific point. And before, nobody really asked whether there is like a moment where the cells can be released actively compared to another timing. And we put in cause, for example, the circadian cycle or the sleep. We don't know exactly. And that's why it's interesting, because we know that also the circadian cycle will be disrupted when you have cancer. So it was from the biology of evolution perspective, but also with the head of narcolepsy in Montpellier, but also is the director and the president of the Narcolepsy Society in French, Yves Dauvilliers. And we ask whether it's sleep or circadian rhythm. And we have some ideas with patients who are really at his department, and we could do that, but we have no time to plan anything. But I would say that it's important, because you can imagine that you have a better way to detect the biomarkers that you need to analyze, but also it was mentioned that you may also treat better the patients where you have dissemination. So at the end, it is the full overview of all the hallmarks of CTCs. And again, I believe that it's very important to understand this biology of the metastatic cascade. And I would like to finish with these specific CTCs at the origin of the next steps of the disease, where the patients will be in a bad position and will die, unfortunately, at the end. So the biology of this subset of cell is very important, and we need to identify the metastasis-competent CTCs. You have different ways to do that, whether you want to directly inject them to immunodeficient mice, or you can try to put them in vitro and try to expand them as spheres. And with my colleague, Laure Carford, we try to expand CTCs from colon cancer patients, 200 patients in this clinical trial. Only one could give rise to a CTC line. And why? Because he had a high number of CTCs in the blood. At least 300, you see, CTCs per 7.5 ml of blood. Otherwise, it was his internal control when you see the progression of the disease, different treatments, different lines, different cures. And you see that you have here the progression. You have a CTC line before treatment, after the first relapse, and just before the patient died. And at the end, it was like growing everywhere. So you see that the pressure of selection with the drug and the different drugs that the patient had made the very aggressive clones growing everywhere. So with the potential to give rise to metastases. And you have here again, you see the number of CTCs detected with the cell search, but you have also the shape and the size. And you see again at the end, you have no clusters anymore and you have smaller cells. So as a sum up, again, for this specific CTC line, what we can say is that compared to primary and metastatic cancer cell lines, not to compare to healthy donors, because otherwise you have too many differences, but just to compare in colon cancer, primary and metastatic size, you have a huge stemness properties for the CTC line, which makes sense because they are able to give rise to new tumor. Then plasticity. So we had a very nice work also with Pelin from Turkey and Mani from US, where we could show that these CTC lines, so we have 9 in total, before, after the first relapse and at the end, that they had an EMT, of course, induced. But you see that the red arrow stopped them very quickly. So they are not really lost in the mesenchymal shape. So it means that you have like a biological lock from these CTC lines that we cannot force to go forwards. When we push them to go in the EMT, it was not possible. So it means that the more aggressive ones are the ones that are able to revert very quickly to give rise to an epithelial shape again very quickly. Then, of course, drug resistance, because they were all still there after such a long time in the patient and after all these drug tests in the patient. And you know this guy who is at the first row. Yeah. Thank you, Daniel, for his collaboration, and Manfred and Klaus. We have this use CTC lines, at least the first one before treatment, to try to understand better and to highlight this specific signaling pathway. And it has been recently also validated in vivo in mouse model. So it's really nice to see that you can use this material, which is very rare for sure, but at least to understand the drug resistance. And we had also a very nice collaboration with Busetter in Harvard, who focused more on the Wnt signaling pathway. So very interesting to use these models because they are replicating like crazy always. Then you see the metabolysis is very important. So we have a strong link with the mitochondria. So it means that they are producing a lot of source of energy, ATP, and so on. And I wanted to let that for the end because we have also in some of the clones the expression of ALDO-B, which is a specific protein of the liver tissue. So it means that these 4 clones obtained at the end are not coming from the primary sites, but they are coming from the liver metastases. So if you have a localized cancer patients, you can be sure that if you see these CTCs, you have already dissemination of tumor cells. But if you have also the expression of ALDO-B, you are sure that you have occurred liver metastases. At the end, Klaus already mentioned this slide, so I go through. But what I would like to show you as a new concept, I would like to share with you because I love this concept a lot. It was just published, but it's the dynamics of liquid biopsy. What it means? It means that we have like the combination, as we said, of different biomarkers that can be a certain type of these biomarkers at the beginning, but it's not just the intensity that we change, that you have more of this biomarker, but there is a change, I believe, during progression of the disease, but also pressure of the drug. And that's why, for me, it's clear that it's stage dependent and cancer dependent, which means that if you highlight in a specific clinical trial that you have this combination for a certain clinical questions, you may have different biomarkers involved in another cancer type or even in the same cancer type, but if you are later in a metastatic disease, your biomarkers will be different. So it makes the situation a bit more complicated, but I believe that it's more close to the truth that in cancer, you know, it's not just one biomarker forever. And for all key clinical questions, I believe that it's changing. And you see, the immune system, of course, will evolve over time. And I hope I can show you more data on that because we have now a very nice signature to discriminate patients with cancer and healthy donors just based on the immune statute. So it should be sent for, I need to read the final version, but it should be sent to the published soon. And of course, here you have the increase of certain biomarkers derived from the tumor, it's obvious, but you have also proteins that might decrease. So you see, it's moving. And if you focus on CTC, they may change as well. You know, you have a phenotype evolving, mutations in the DNA and so on. So it means that it's really the dynamic of liquid biopsy. So what I believe that we need more interventional trials, like Klaus said. It's very important to have to show that you can change the management of cancer patients based on the biology and liquid biopsy, whatever the biomarker will be. And that's why I would like to highlight that this one, the first one in metastatic breast cancer. And I will have a huge meeting in December in front of the High Authority of Health to push maybe more in the direction to be reimbursed in France, KISS. And of course, you see that I believe really that we need to combine different biomarkers induced by the tumor, which is new, not only derived from the tumor, because something is happening before you have a tumor cells, I'm sure of that. So for early detection and PANCAID, I believe that we have to also combine things before. And that's why we have the right cohorts of patients, maybe to achieve that. Of course, as we have a lot of information, I lead the group of Madrid now and Noya's group, which is very important, artificial intelligence. And I was happy to be interviewed by Nature, and they highlighted our other project, Biopsy on early detection of pancreatic cancer and Liquid Biopsy, how artificial intelligence is helping to boost cancer screening. And I believe that it will be possible in the future, for sure. Then at the end, Klaus mentioned ELBS, which is important to work all together in the same direction. Otherwise, we have so different way to work on liquid biopsy that it's difficult to compare the data. And then I would like to say as well, they are not still there. That's why I asked Tamara whether they will be there. But it's nice to see also that we can interact. And there, I was invited to give also a teaching lesson for 1 hour in Uppsala University. And of course, we highlighted all the work on early detection in PANCAID of pancreatic cancer. But it's nice to see also that the different clusters can meet not only in the PANCAID, I would say, meeting. So thank you for your attention. And if you have questions to me and Klaus, I'm very happy.